Hello and welcome to today's webinar. This is Peter Behor from the U.S. Environmental Protection Agency in the Office of Compliance. My colleague Laura Paradise is operating the webinar. Welcome to today's webinar, the microbiology of wastewater treatment, life in the aeration tank, bacteria, protozoa, and metazoa. Today's webinar will introduce the audience to the microorganisms that thrive in activated sludge. Today we are lucky to have John Van Domelen. This is John's fourth time presenting. John has worked with or for EPA, Ohio EPA for 25 years. He spent the first 6 years reviewing detailed plans and issuing permits to install for new wastewater treatment plants and collection systems in the Division of Environmental and Financial Assistance. He eventually transferred to Division of Surface Water, joining the Compliance Assistance Unit. 4 years ago, he returned to DEFA along with CAU. He has worked in the CAU for a total of 19 years, troubleshooting noncompliance at wastewater treatment plants ranging from 1,500 gallons per day package plants to 15 million gallons per day municipal systems. In addition, John has trained operators of wastewater treatment plants in process control and activated sludge microbiology, biological nutrient removal, among other topics related to wastewater treatment. He has also presented at WEFTEC 3 times at many OTCO and OWEA workshops, as well as other regional, state, and local organizations. He has published articles pertaining to topics of interest to operators in Buckeye Bulletin, publication of Ohio Water Environmental Association. John graduated from Ohio State University with a Bachelor of Science in Civil Engineering and is a registered professional engineer in the state of Ohio. At this point, I'm going to turn it over to you, John. And
okay.
Thank you for helping us once again.
My pleasure. Thank you, Peter. Thanks for the great introduction. Okay, so like Peter said, this is my fourth webinar for the US CPA webinar series. Maybe you've heard me before, maybe not. Anyway, this is one of my favorite presentations to give. I give it frequently, and there's something about microbiology and looking under a microscope that just works for me. I'm never bored when I'm looking under a microscope. So we have for about the next hour and a half, we should hopefully you will gain some information and hope you'll find it useful. So there's my photo. I was reluctant to do this, but I thought, well, I'm not gonna put my web camera on. But anyway, I work for Ohio EPA in the Compliance Assistance Unit. And what we do in the Compliance Assistance Unit is we go to treatment plants that are having compliance issues. We go to the plants, we work with the operators to figure out why they're having a compliance issue, a non-compliance issue, and then we work with them to correct whatever it is that's wrong to get them back into compliance, okay? So I've visited probably hundreds of treatment plants in the 20 years that I've been doing this job. And again, it's gratifying because the operators that I work with, they are usually so appreciative because they've tried all sorts of things. And when we come in, we've seen so much non-compliance that we've gotten pretty good at pretty much figuring it out. And we have great tools, but one of those tools that I do use frequently is the microscope. So let's get down to business here. Why does my microscope work better than yours? That is, if you don't have a phase contrast microscope. Well, there's a very good reason for that. Right there is a couple of my microscopes. The one on the left is a phase contrast. It's a lesser microscope, still about $4,000 microscope. But the one on the right is my baby. That's my and the small one we loan out to operators occasionally. The one on the right is my baby. That's the one that I carry with me. It's a really nice microscope, and it's phase contrast, and it has a trinocular head that has a camera on it. And the camera will display on that little notebook type display. And I can even attach a cable to that display and run it through a data projector, so I can do this for large groups. I can put it in the, it has an Ethernet connection too, so I could actually display what's underneath the microscope to a wide audience. Okay, so anyway, those are my microscopes that I use. And this is a picture of underneath my microscope, only this is a bright light setting rather than the phase contrast setting, just to show you why a phase contrast microscope is really what you need to do filament identification, as well as protozoa identification and flock identification for that matter. So this is a bright light setting, which I have on my phase contrast, and the exact same image with a phase contrast setting looks like this. Pretty dramatic, huh? Okay, you can see, well, let's go to the next one. Again, now this is, that first one was about a, I don't know, 100 magnification. This is probably about a 400 magnification. And again, you can see that it's a stoxiliate, and you can see some filament winding through there, but you can't see any details. I flick the switch to phase contrast, and voila, there you go. Much different image, correct? And then here are some filaments. Again, bright light and phase contrast. Okay, so these are all photos that I've taken with my camera, with my microscope camera. And actually, every photo in this presentation are my photos that I've taken just from years and years and years of taking photos and looking at mixed liquor samples under the microscope. So here's a typical microscope. This is like my small one, my loaner, and it just shows how the light goes up through the optics of the microscope. But the phase contrast microscope has these special features. It has a condenser ring, which is underneath the stage where the slide sits, and it has a ring aperture in the condenser. There's one for each of the microscope objective lenses. And then in the lens, you have a phase plate, okay? And just these two little devices give you that depth and clarity that a phase contrast microscope will give you, okay? And really, to do any kind of filament identification, you really need a phase contrast microscope. So if you have one, great. If you don't have one, then hopefully, not everyone can afford a $4,000 microscope or an $8,000 microscope. But I don't know. I take this microscope to small systems where operators have never seen what's in their tanks, and I show it to them, and they're just always amazed, as am I. So, okay. So, why perform a microscopic analysis? That's a very good question. One, if you know what it looks like under the microscope when your plant is running really, really well, then you can almost always, if you're doing a microscopic examination on a regular basis, you can almost always see changes in the samples under the microscope before they become problems, okay? For instance, there are, as you will see, and look at my background of my slides here, you see flock and filament, okay? If you see nothing but flock, then it probably settles well, and you probably are treating well. If you start to see filaments coming out of that flock, or filaments in the bulk liquid, like the background of my slide here, then before it gets too bad and causes you a settling issue, you get that preview, and you might be able to change the conditions in your tank in order to select against that filament. Okay, so that's one good reason. Also, the protozoan that you see, same thing. If you see certain protozoan and you're looking at it again and all the protozoan are dead, well, that's telling you that there's something toxic maybe happening in your aeration tank. And you might not know it if you're sending your samples out, your effluent monitoring samples out to a lab, you get them back 10 days later or so, or if you're doing your lab work, every twice a week or once a week or something like that, it might be too late. These are the essential resources. The book on the left by Dick Eichelboom, Process Control of Activated Sludge Plants by Microscopic Investigation. That's a great plant. Dick Eichelboom is a giant in the wastewater microscopy world. I believe he was the guy that named some of the filaments type and then a number, like type 021N or type 0040, something like that, or type. Well, anyway, he was the guy that named those. The book on the right is by Toni Glimpf. She used to work in Wisconsin. I believe she's retired now. And Toni and she taught me a lot about microscopic examination. She's an honest to God microbiologist. And she and I, in this book, sat down. We got together. She came to Ohio. We went to a lab in a bigger wastewater treatment plant, and we collected samples from the nearby treatment systems. And we sat there for 2 days and took photos of the biology, anything interesting that we found, and she used those in her book. So I'm proud to say that about 75% of the photos in her book are my photos. And then the book in the middle, the manual on the causes and control of activated sludge bulking, foaming, and other solid separation problems by David Jenkins, Michael Richard, and Glenn Dagger. This is my really go-to book because there's so much information in this book. And it's when I have a question about solid settling issues, solids problems, this is usually the book I go to, but I look at the other ones as well. But these three, I highly recommend any of them because they're excellent resources. Actually, I'd recommend all three. Okay, so first off, we're going to look at flock structure. Do you guys want to do a polling question here?
Sure, John. I
Okay.
will bring up the number of how many years of experience. So you guys will see on your screen here, I'm going to launch how many years of experience people have. So you should be seeing this on your screen. If you would click on what's appropriate for you, we'll give you about 20 seconds or less to respond, and then we'll post it out to everybody. Give you about another 5 seconds. 4, 3, 2, 1. We've got about, okay, I'm going to close it. We've got about 40% of the people. I'm waiting to 50. All right, closing it. Got about 55% of the people vote, and I'm going to share this with everybody so we can see. You'll see that 50%. John, I don't think you're able to see this when we post it. Yeah, so we have 0 to 5 years. We have 50% of the audience. And 0 to 10, we have 14%, 11 to 15 years, 9%, and 16 and more years, 27%.
Okay.
So.
Well, good. That's good. I'm always curious to find out who's attending. So that's good. We got both people that are new and people that are experienced. And hopefully I can teach something to all of you. So, okay, flock structure. This is one of the important things that you can determine under a microscope. Okay, here is dispersed bacteria. Now this happens when you have a lot of food and not very much bacteria. If this were a live microscopic image, all that bacteria would just be sort of vibrating back and forth, okay? This tells me that, you know, there was maybe a washout or maybe a new plant just getting started. But anyway, there's all this bacteria and there's a whole lot of food, a whole lot of BOD, okay, and ammonia and such. And the bacteria doesn't flocculate when there's that much, when there's that few bacteria and that much food. But eventually, if you give it enough air and keep it warm enough, you will start, the bacteria will start to flocculate. And this is the beginning of flocculation. You can sort of see the pattern. What happens is when the food gets, starts to begin to get scarce, the bacteria will start forming slime on their cell exteriors, and those slimes will stick together when they collide. And this is a photo of a monoculture, and it was just beginning flocculation, okay? Here, this is probably from a package plant. They're very small flocks. Package plants are usually low-loaded systems and usually don't get a lot of attention, and they usually have pretty violent aeration. And so you end up growing small flocks because there's not a whole lot of food coming in, or it all comes in say like an 8-hour period, and then the rest 16 hours, it's maybe not very much food coming in. And so they grow very small flocks. And some of the bigger of these small flocks will settle pretty well. Some of the smaller ones will not settle very well, and that will look like turbidity in a settleometer or in your clarifier. But under a microscope, it just looks like very small flocks, okay? You can see it's everywhere. Now this is a big dense flocks. Now what you see here is you see these larger flocks, and you don't see all the small flock in the bulk liquid around it, okay? And you can see a little bit of filament, little hair-like structures coming out of this these compact dense flocks, because the bacteria, the flocculating bacteria, will form around the filamentous bacteria. So a little filament's not a bad thing, okay? And this will probably settle very well and compact very well. And you can see that just in the difference between the small flocks and the dense compact flocks, okay?
Hey, John, I have a quick question for you.
Sure.
Do you know the magnification rate that you have these slides?
Yeah, this one's probably 100 or 200 magnification. This one is probably 100 to 200 magnification. This one is probably 1,000 magnification. Probably should have put little magnification bars on that at the bottom of each of these. I'll do that the next time. But anyway, these are flocks. Now you can see inside the flock here, this is a close-up, 1,000 magnification of the inside of a flock. And what you can see is you can see the filaments. That's the spaghetti-like structures all through this, view. And it actually gives that flock a little bit of structure and form, kind of like the old plaster, house plaster that had horsehair in it. You know, it had fibers in it, so it would add strength to it. But this is fine as long as the filament stays under control. If the filament starts expanding and growing, then you get something like this. This is probably about a 200 magnification. What this is, you can see the flock. Some of them are pretty dense. Some of them are a little spread out, and you can see all the filament connecting all those flocks. And this is a problem because when you have that much filament and what we call interfloc bridging in a settleometer or in your clarifier, the filament will keep those flocks from compacting and forming a good settling blanket. But it will if there's no heavy flow coming in at the time, or if you have an influent pump station, the pump station's not pumping. A sludge like this will filter really, really well. And so you can see crystal clear effluent on top, but say a pump kicks on, and because all these individual flocks are connected by that filament, and that creates a low center of gravity. And so if the pump kicks on, then that's likely to escape the clarifier, go over the weir, and either clog your sand filter or go out into the receiving stream. Okay. So interfloc bridging is a real problem. It can give you crystal clear effluent if you can keep it in your plant. The next one, again, you see a little difference here. You see a lot of floc and some filament, but you don't see very much bulk liquid, okay? This is just too much mass. Now you can have settling issues if you have way too much mass and not having, and don't have any filaments, or none than the normal amount of filaments that you might have. But with a mixed liquor like this, there just isn't any room for it to compact much further. So in this case, waste, okay? And then you have this one. There's a lot of floc and there is a way too much filament. This is almost all filament right here. So this was a very particularly bad mixed liquor that we had in the treatment plant. But anyway, too much filament. Okay, now we'll go into protozoa and metazoa. I think we'll hold off on a polling question here and we'll do it in the next one. Now, these are other organisms that live in the aeration tanks other than the bacteria, the floc and the floc forming bacteria and the filamentous bacteria. Protozoans are single-celled organisms and metazoan are multi-celled organisms. And they're there because their food sources are there. Okay, let's take a look at some of these. Okay, you're liable to see any of these anytime in any view, but what we're actually looking for is what is there a lot of. These are amoebas. Okay, the top one and the one on the right are what they call naked amoebas. They don't have much structure. they have some structure, but they're mostly just sort of amorphous globs. But the one in the bottom left is a testate amoeba or shelled amoeba. The shells are called tests, and kind of looks like a Cheerio right there. But the thing with amoebas are, at least the naked amoebas, is they expend a lot of energy when they are looking for food. Because when they look for food, they're out just kind of moving their protoplasm around, and they'll find a piece of food, and then they'll wrap their cell structure around it, and then it'll absorb it into the amoeba, okay? But the testate amoeba, the shelled amoeba, is usually seen in well nitrifying mixed liquors. So, and they don't move really. They are pretty stable. So, but they're a good indicator because if you see a lot of shelled amoebas, you know that they had low ammonias for quite a long time. If you see a lot of dead shelled amoebas, gray ones instead of brown ones, then you know they had low ammonias for a long time. And then something happened and they probably had high ammonias and it killed the shelled amoebas. Okay, it's one of the things you can learn by looking under a microscope. These are flagellates. Flagellates are hard to photograph because they move around very, very quickly. You see them buzzing all through slides. And when you put them on 1,000 power, the focal length is very narrow, and it's hard to get them in focus. And so anyway, they're hard to capture, but I did manage to capture some of these, and these are all flagellates. And again, they move around, they're looking for food, and they expend a lot of energy looking for food. So if you see a lot of flagellates looking around or zipping around, you know that maybe your treatment isn't really all that great. You might have some higher ammonias, might have some BOD present. There's one really big, slow-moving flagellate, though, that shows up in well-treated mixed liquor, though. But these little ones, the little tiny ones, usually indicate that you're not treating as well as you should. Okay, the next thing on the food scale, on the critter scale here, is the crawling ciliates. Now these guys, you'll see these crawling on the flock, and they're ciliated protozoan, okay? And but they do have these specialized cilia. The one on the right, especially, it looks like it has 3 little legs right down there. They're not legs, they're cilia, but they're specialized cilia, and they do use them for mobility. You often see them crawling around the flock looking for some particulate BOD to eat, and you know, some of them look like, if you get them in a different view, they look kind of like a helmet. But anyway, these are often visible, just crawling around, even on well-treated mixed liquor. Okay, so now we're getting towards a little bit better treatment. These are stock ciliates. Now, stock ciliates don't move; they're stationary. Okay. But they do have cilia around their mouths. The one on the right is a pretty good view of the circular opening on a stock ciliate. Again, this is a single-celled organism. And those cilia will rotate in such a manner as to create a vortex that will draw water into the mouth of the stock ciliate. And they can process a lot of water, because there's not a whole lot of food left, because the treatment is getting much better. So they will take food in, and then eventually they can release it. I have a couple different varieties here. I think the one on the right is Vorticella, and the other 2 are Opicularia, I believe. Okay. And then I have this one photo. I had a brand new camera for my microscope, and one of the very first pictures I took was this one. And it just so happened, this is one that has a contractile muscle in the stalk of the ciliate. And as I took the picture, it contracted, and got that Doppler shift there, the blue-red shift of the movement. And actually, at the Ohio EPA, when we moved into a new building, they redid a huge department store in downtown Columbus, and the agency moved into the top 3 floors. And when they rehabbed the whole building, they had all these blank walls in the offices, in the hallways. And so they had a photo contest for the employees. And I thought, okay, I'll submit some microbiology photos. And of course, no one else did. And they were so impressed with the photos that they created a new category, and they actually printed 10 of my photos, had them professionally printed and framed and labeled. And when you got off the elevator at the reception area at the agency, and the elevator doors would open, you'd see 3 of my photos right there. First thing you would see is the agency. So I was rather proud of that. They had a couple others throughout the building, but this was one of those ones that was in the lobby of the reception area. Okay, so here's another one. This is sort of a cousin to the stock ciliate. It's a single-celled ciliated protozoa. It's called Vaginicola, and what it does is it secretes this shell. You can see it in all three images, and it lives within the cell. Sometimes there's one organism in a cell. Sometimes there's two organisms in the cell. But what they do is they'll come out and feed, and then they'll retract and go back in, and then they'll slowly come out, kind of like seeing if the coast is clear, and then they'll start using their cilia to create that vortex to bring food to them. These are another organism that is very sensitive to ammonia. So if you see a lot of these and they're active, you know that they've had low ammonias for a long time, okay? Or else they wouldn't be there. Or if you see a lot of them filled with bacteria instead of the ciliated protozoa, then you know that they had low ammonias for a long time, but something happened and they all died, okay? You know, if I were an inspector, which I'm not, I'd probably carry a microscope around and look at the mixed liquor, probably tell me exactly what's going on in the plant. Okay, let's look at the next one. This is a good one. This is a Suctorian. This is better than science fiction. Now these guys they're kind of like a stock ciliate, only their cilia are spikes. These are straight, hollow spikes. And what they do is they wait for a protozoa to come by and bump up against it. And when they bump up against it like that amoeba up there, then the Suctorian will secrete enzymes into the amoeba, or whatever protozoa gets stuck to it, and it will digest it there, and then it will suck its contents out again, okay? Pretty violent, huh? I've only seen one time when a protozoa got stuck onto a Suctorian, and I was doing a microscope show for my daughter's second grade class. I had the microscope cameras set up to a TV set, and sure enough, there was a Suctorian in the image, and a protozoa got stuck on it, and I couldn't snap a picture because I didn't have that camera on it. I had the video camera on it, but not the still camera, and I missed it. I've never seen it since. But that's what they look like. Yeah, it's pretty interesting. Looking under the microscope, you get all sorts of interesting examples of this life in the aeration tank. Okay, the next thing, now these are metazoan. These are multicelled organisms. This is a rotifer, everybody's favorite. They're kind of charismatic. They're big. They're active. They all have two little toes at the bottom end, and they both have they all have two mouths, one on each side of their head. And if you look sort of in the upper third in the middle, you can see a little structure in both of these. The one on the left's active, the one on the right is probably not very happy and might even be dead. But what it looks like a heart that beats, but it's not really a heart. It's really its jaws. So it brings food in through its mouth, and then it's like a grinder. That's how it chews up its food, or its, whatever it eats, the bacteria and such. Okay. But the other thing about rotifers is they are all female, apparently. The males are short-lived, much smaller, and they die shortly after fertilizing the eggs. So anyway, those are rotifers. Very interesting to watch. These are nematodes. These are worms, microscopic worms. Now, Mike Richard, who was one of the co-authors on the Jenkins Manual, the book in the middle, I took a class with him out in Oregon, 2.5-day class, where he brought in every filament that you would possibly see in a mixed liquor situation. And the one thing he said about nematodes was, if you see nematodes in your mixed liquor, congratulations, you're no longer activated sludge, but you're liquid compost. He said, worms, nematodes are compost organisms. So you've gone from mixed liquor to, or you've gone from activated sludge to liquid compost. That's a little dramatic, but still, it's interesting. I see a lot of nematodes in people's mixed liquor, and it usually tells me that they probably aren't wasting quite enough as they should. This is another worm. This is a bristleworm. Good story going along with this one. This bristleworm, it's huge. You can almost see these with your naked eye sometimes, but you know, in mixed liquor, it's kind of hard to, it's not very clear, so you can't really see them individually. But under a microscope, you can see their entire digestive tract contracting and such, because they're pretty much transparent. And the other thing is they have these little photoreceptors in addition to the bristles coming off of them, which is where they get their bristleworm name. But they have these little dots all over their body, and they're photoreceptors. And one time I was at a wastewater treatment plant and it wasn't settling very well. And I did a settleometer and I set it up in the windowsill. And then I was going about doing some chemical analysis. And when I went back to my settleometer, it didn't settle very much, but the whole side towards the window was kind of reddish. And I thought, wow, that's weird. So I had my microscope with me and I took it out and looked at it, and it was full of bristleworms. And those photoreceptors gravitated towards the sun, which was in the open window. And that was, you know, it's neither here nor there, but it was an interesting realization that these things are attracted to the sun. You can see a couple other things here in this photo. Up in the upper left-hand corner, you see a testate amoeba kind of on its side, and you see a vaginicola down at the bottom right, or bottom left. So see, all these things are in these tanks. So low ammonias in a higher organism in good dense flocks. Okay. This is a paramecium. Saw this, you don't see these very often, but you see them occasionally. You can see they're covered with cilia. I know, took a photo of this one, and just because I saw it, and I've seen a few of these, but not very much. They're pretty much freshwater organisms, so you don't see them very much in wastewater treatment plant mixed liquors. This is the stentor. This is also a single-celled organism, kind of like a stoxiliate, kind of related to it. It's kind of like the vaginicola without the shell. But this one came from, I was doing a micro show for a high school science class, and the teacher had, it was a rural high school, and the teacher had the students bring in samples if they had, you know, standing water around someplace. And this sample came from a manure lagoon in a farm that hadn't been used for like 30 years. And he took a sample, brought it in, we put a drop under the microscope, and there were tons of stentors in it. And I've seen this a couple times in mixed liquor, but not very often. Anyway, you can see those as well. And then there's the tardigrade or water bear. These are fun to watch. These are, again, kind of like better than science fiction, because these guys have 8 legs. You can see 4 of them on the left-hand side, and on the other right-hand side, 3 of them are underneath his body. but they have 8 legs, and at the end of each leg are these big claws, like a bear claw. That's why they call them water bears. And these guys are, you know, almost indestructible, I guess. I mean, they can ball up and survive drying out and all sorts of things. You know, they find them on nuclear waste sites and such. But these guys are real interesting, and they just go crazy underneath the slide. They just go scooting all around usually. Sometimes you see pregnant ones. They have eggs inside of their bodies, and sometimes I've only one time seen the baby water bears developing inside the egg cysts, but they're pretty interesting too. Again, this is a metazoan, multicelled organism. Okay, so that's pretty much what you would see as far as protozoa and metazoa. And this is a chart from the Eichelboom. And what this does is it shows you where these organisms grow in mixed liquor, okay? So you can see there's 2 main groups. The bottom 2, the flagellates and the amoebas, grow way out where there's a lot of food. Okay, so in F to M, they have numbers here. I don't like using numbers for F to M because, you know, well, I have my own reasons for that. But you either have a low F to M environment where you have very little food and a lot of mass, or you have a high F to M environment where you have a lot of food and not as much mass. So if there's plenty of food, you'll see amoebas and flagellates and some free swimmers, okay? But then once the food starts getting scarce, everything shifts to the left, to the low F to M environment, and that's where you see the crawlers, or more particular, the stalks, the carnivores, and the shelled amoebas, okay? Those are the protozoa that will survive in a well-treated mixed liquor, okay? And the metazoans as well. They will live because a lot of the metazoans are very sensitive to ammonia toxicity, okay? Okay, so that's it for the protozoans. So now we'll look at filamentous bacteria. And if you have another polling question, let's go with the next polling question.
Yep, I was waiting for you, John. Let's see, I'll launch the next one. And does your facility have use of a phase contrast microscope? Yes or no? So if you guys would all see what percentage we can get of the group. We currently have about 1,080 people attending today. So it'd be nice to get at least half of you voted. And we've got only 11% so far. And it's growing quickly. So once we reach the threshold there, we've got about 42%. And I'll give you another 5 seconds. 4, 3, 2, 1. And I'm going to close the polling and see him. And now I will share it. So John, of the 58% that responded, 68% said no, they do not. And the remaining 32% said yes, they do.
Oh, that's great. That's good that there are that many phase contrast microscopes out there. I'm impressed. So I guess my microscope didn't work any better than some of theirs.
Do you want to push out the next one later?
Yeah.
Okay.
After the filamentous bacteria section is over. Yeah, that's pretty impressive that there are that many people with phase contrast microscopes. There are good phase contrast microscopes and there are lesser phase contrast microscopes. be honest with you, the cheaper ones don't have as good of optics, and the more you can spend, the better the optics are. So anyway, that's that. So now we're going to look at the filamentous bacteria. Now this is something that is what I really use my microscope for, because if you're having pore settling sludge, a microscope can tell you not only whether it's filamentous or if you just have too much mass, but it will also tell you a little bit about what the conditions are in your aeration tank. Filamentous bacteria grow under very specific environments in the aeration tank, okay? There's what I call a low F to M environment, meaning that you have way more mass, way more bacteria than you have supporting, than the food that's coming in will support, okay? Or their filaments grow under a low DO condition, a chronically low DO condition, okay? So in those two instances, the filaments will outcompete the flock formers when nutrients get scarce, okay? So if the food is scarce, the filaments will have more surface area than the flock formers that form into a ball or a flock, and they will outcompete for scarce resources. And oxygen is another scarce resource that the filaments will outcompete the flock formers for. Now, there's other filaments that actually grow on oil and grease. That's their food source. But these filaments are hydrophobic, meaning they don't get along with water, so they float, okay? Because oil and grease will float in a water environment, and it goes to the surface. The bacteria will go to the surface because they're hydrophobic, and then when you aerate it, you get lots of foam, okay? Lots of foam. You can get lots of foam. Then the third one, or the fourth one, that happens is the products of septicity. When I talk about septicity, it's like that first stage of anaerobic digestion, the acid forming stage. And when sludge and biology and such start breaking down, that first stage of anaerobic digestion is the acid forming stage. And what they form is low molecular weight organic acids, okay? And those low molecular weight organic acids, you put those into an aerobic environment with lots of DO, and the septicity filaments, it's like candy for the septicity filaments. They will just take off, okay? And then the other condition, which you don't really see very much in a municipal treatment systems, but you can see it in industrial treatment systems, is a nutrient deficiency. And these are the same filaments that form in those other categories, but they can also get a competitive advantage when there's a major nutrient that is deficient, like phosphorus or nitrogen, okay? A classic example would be like a cheese house, cheese manufacturer. Cheese waste is deficient in nitrogen, and it has lots of phosphorus. So you get an imbalance in the nutrients, and you can grow a nutrient deficient sludge. And the two main ones that you see are if you have a nutrient or a nitrogen deficiency or a phosphorus deficiency. And usually with the nitrogen deficiency, you will grow a prolific slime on the outside because they can't metabolize their slime layer, and the slime layer will go haywire, and you'll get all sorts of slime bulking if you have a nutrient deficiency. If phosphorus deficiency, you can get the same thing, only to a different, slightly lesser degree. But anyway, these are the two usually that give you slime bulking, okay? And I had a question come in ahead of the presentation, just talking about a membrane bioreactor system that the membranes were getting clogged with slime, nutrient slime. And so probably if you should be, in that particular case, they should be measuring orthophosphate or ammonia or nitrate to find out which one is at a, which one's deficient or which one is not really very, not present in the reactor, okay? So anyway, these are the filamentous bacteria that are commonly found in wastewater treatment plants, and they're by category. You have 4 low F to M ones, low, too much mass for the amount of food coming in filaments that will take off under that environment. There are 3 that will do oil and grease. There's 3 that are responsible for or will take off it in a low DO environment. And then there's a whole host of them that will respond to septicity, okay? And then when you get down to nutrient deficiency, those are all filaments that are in the other categories, but can also proliferate when you have a nitrogen deficiency or a phosphorus deficiency, okay? So this is the main reason why you do a filamentous or a microscopic examination of your mixed liquor. Filaments don't lie. If you can identify the dominant filament, then you can go into one of those reference books and identify the growth condition in that reactor. Low FDM, low DO, septicity, nutrient deficient. And then you can change the conditions in the reactor. Waste more, waste less, turn the DO up, turn the DO down, you know, add a nutrient if it's missing, or eliminate the septicity. And if you change the condition in the reactor, you will eliminate the problem, okay? That's the whole reason behind doing microscopic examination for poor settling sludge, okay? So here, we're gonna look through the main filaments that I see in mixed liquor. And where I have photos, I'll have a wet mount on top. In the lower right-hand corner, or left-hand corner, we'll have a gram stain. And in the lower right-hand corner, we will have a Neisser stain, okay? Where applicable. Not all, well, this one, type 0041, doesn't stain. It's a Neisser negative filament, so I probably wouldn't take a picture of that and send it to a client that was having an issue. But anyway, this is the first slide of the filaments. So anyway, these are the filaments. This is type 0041, okay? As you can see in the wet mount, it's a pretty robust filament. That's 1,000 power. Relatively square cells. You can see the individual bacteria cells, and you can see that it grows in a sheath, because at the very end on the right-hand side, you can see the empty sheath, okay? And the other identifying feature of this type 0041 is the fact that it has lots of attached growth. You can see it in the wet mount, and you can see it in the gram stain. It's gram positive, these are negative, and you can see the gram negative, they call it epiphyte, but we call it attached growth, colonizing the sheath of the type 0041, okay? So what are the growth conditions? This is your classic low F to M environment growth condition. And it grows on slowly degradable or particulate BOD. And if you have a lot of it, you need to increase wasting, okay? I don't really see a whole lot of sludge bulking due to type 0041 because it's usually in the interior of the flock and it will extend out a little bit. And it's a particularly hardy filament, but usually you see it in almost every sample, and it doesn't really give you a whole lot of pore settling, but it can if it really takes off. Here's another one, no Neisser stain here, but this is type 1851. This is another low FDM filament. It's identifiable. One, it's very thin, and it usually has just a little bit of attached growth coming off at a right angle to the filament in the wet mount up above. The other thing with type 1851 is that it is typically grows in bundles. Like that's a pretty extreme bundle at the bottom. It usually looks more like the wet mount, but it really does grow in bundles, and it's kind of weakly gram positive. It's not that really dark purple like type 0041, but it's sort of a weak gram positive reaction. Again, this is a classic low F/M filament. You often see it in completely mixed basins, completely mixed aeration tanks instead of a plug flow system. A lot of times the complete mix might have a mechanical mixer in the middle, and flow comes in, it gets dispersed immediately. And so even though it might be a high F/M, it gets dispersed immediately, and to the bacteria, it looks like a low F/M situation. Classic response for low F/M filaments, again, is increase wasting. Or if you can change your configuration to a plug flow configuration where you have a high F/M environment at the beginning, and then as you move on, it gets to a lower F/M. Or you can do a selector, usually like an anoxic selector, which gives the flock formers the first kick at the soluble food, okay? And then you can let the flock formers proliferate first, feed first, okay? This next one is Microthrix parvicella. The top one is a wet mount. Again, you can't see the individual bacteria cells. It's a coiled filament. It doesn't have attached growth, and it's strongly gram positive. That's the lower left-hand side. It looks like a plate of purple spaghetti to me. So, and the flock that's around it's just incidental, but it's not attached to the filament itself. And the Neisser stain is interesting because nothing else looks like that when you do a Neisser stain. It's not a true Neisser stain reaction because a true Neisser stain, the body of the filament will stain deep purple. But here, just there are little granules inside the filament that stain deep purple, but the filament itself stains kind of that golden color. And there's really nothing else that looks like that, okay? So that's your primary identification by doing a Neisser stain. If I suspect parvicella, I'll do the Neisser stain first, because there's some things that could look like the gram stain, okay? Okay, this is one of the oil and grease ones. Microthrix is actually grows on oil and grease, and there's high carbon chain fatty acids, but it also grows at a low F/M, and it can grow where you have intermediate low DOs. Okay, so it's kind of like the triumvirate there. A little bit of everything supports Microthrix's foothold in the mixed liquor. The literature always says it tends to favor cold water temperature, but you know, I see it summertime, wintertime, I see it all the time. So I used to see it a whole heck of a lot back before people had bigger digesters. Back when I first started in this position, we saw microthrix everywhere in oxidation ditches, all sorts of plants. And as people started doing upgrades in the mid to late 1990s, then they started wasting more, belt presses became more common, they started handling their solids better, and they weren't holding on to their mass so long. So anyway, proper response, oil and grease control. Now you don't have to have excessive oil and grease, you just have to have some oil and grease. Bigger plants, the primary clarifiers tend not to have Microthrix parvicella because the primary clarifiers remove the floatables, remove the grease. Foam trapping, okay, because parvicella floats and you aerate the mixed liquor, that creates foam, okay? When you aerate bacteria, it will foam if they're floating. And if you trap the foam with, say, a subsurface takeoff rather than overflow weir takeoff from the aeration tank, it will, all that foam will stay in play the whole time at that water foam interface. The stuff on top, a couple inches up, might dry out and might be dead, but right there at that foam water interface, it's always in play. So if you're going to get rid of it, you have to suck all the foam off, okay? And increase wasting. That's usually the main thing. If you keep on top of your sludge inventory, you shouldn't have parvicella. And maintain adequate DO, okay? That's the other one. Okay. This is Nocardia. This is another one that gives you lots and lots of foam. Nocardia is, again, gram-positive, and it has a pretty unique Neisser negative, but the granules are Neisser positive. But unlike parvicella, the body of the filament is pretty transparent and slightly Neisser positive, but the granules are very Neisser positive. Nocardia is one of those filaments that everybody seems to have a little bit of. As you can see in the wet mount at the top, it has true branching, and but as you can see, the filament itself is very short, and it usually doesn't give you a settling problem unless you also have a whole lot of mass, and the mass will give you the settling problem, and the Nocardia will just be there because of having too much mass, actually. And the oil and grease, but this one also feeds on oil and grease. I have had some big experiences with Nocardia. Here's the conditions it grows under: fats, oil, and greases. Okay, foam trapping again. Lower F to M environment again, and this one, low aeration tank pH. It really, really likes a low pH. In the response to get rid of it, oil and grease control, primary clarifiers. You don't get Nocardia either. You don't trap the foam. You increase the tank pH. Now that's easy to do in a small system just by adding sodium bicarb to increase the alkalinity. Big plants, it's a little bit more difficult. So, you know, if you have primary clarifiers, that's a better way. If you don't, if you can create an anoxic environment to increase alkalinity, that can raise pH. And the other thing is you have to waste a lot, okay? So here is Nocardia foam. This is a plant that I was called to. They discharge their effluent to an impoundment, and they use it to water a golf course. but they had Nocardia so bad. They had like 2 to 3 feet of Nocardia on top of their aeration tanks. You can see the top of the tank in the background there, and it's coming out of the top of the tank in the foreground. And that's probably a good 2 feet of foam on top of that aeration tank. Okay. This is the page out of the manual on the causes and control of activated sludge bulking, the Jenkins manual. And what this shows you is a real interesting situation. This is back in the narrative part. We'll talk about the filaments per se, but they have a whole section on Nocardia. And, you know, I used to overlook that part of that manual frequently because I'm always looking at the filamented part. But when I got called to that project with all the foam, I started reading the manual in the back part, and I found this chart. And what the chart says, they have an experiment, and the y-axis is the amount of Nocardia filaments. I probably had a graduate student count it. And you can see it goes from 0 on up to, I don't know, about 12 times, be 12 million per grams of volatile suspended solids. But there's foam trapping in that first section. And then when they get rid of the foam trapping, it starts coming down. And those are both under a 10-day MCRT, mean cell residence time. Okay, so then they go to a 5-day MCRT and it stays down as long as there's no foam trapping. But then, you know, I don't know what they did, but they started trapping foam again. I don't know why, but they did. And look, the Nocardia took off again. Okay, so foam trapping is one of the major causes of excess Nocardia. Okay, and here's another chart from that same manual. And what this one too shows me is the low pH. So you have two plots here. The solid plot at the bottom is a 3-day MCRT. Now, not many plants have a 3-day MCRT because you probably aren't nitrifying at a 3-day MCRT. And in Ohio, almost everybody has a ammonia limit, so you need to nitrify. But at 8-day MCRT, yeah, you probably nitrify at 8 days. But look at the growth rate, the slope. Okay, so you have your upward slope between about 6 and about 6.2. That means that the Nocardia grew at a very fast rate. Between 6.25 and 6.5, it's still going upward. So when you're right around that 6.5 pH, you're still growing Nocardia. Once you get above 6.5, it starts coming down a little bit, but until you get up to about 7, it only comes down quickly. It comes down more rapidly at a faster rate, okay? So those two things, foam trapping and pH, because that plant that had all the Nocardia foam didn't discharge to the waters of the state, but discharged to an impoundment, they didn't have like a low pH in their permit because it was going to be reused. So their pH was usually right around 6.5. Sometimes it would be less, sometimes it would be more. And that's why they grew so much Nocardia, because it grows under low pH. And then if you trap that foam, it just makes it worse. So, and as you can see here on this chart, again from the same manual, Nocardia grows from anywhere from about a 2.2 MCRT on up to infinity, I guess. So it grows under a wide range of MCRTs. There is the same treatment plant after we, 1, created an overflow weir so there wouldn't be foam trapping. 2, it was a 3-pass system we got rid of one pass and just did it down and back. Well, they had a 3-pass system that ran in parallel. But what we ended up doing was running in plug flow in 2 passes down and back, and wasted a ton of sludge, sucked a ton of foam off the top of the layer. The street department came in and sucked foam for 2 days. And then they started adding sodium bicarbonate to the mixed liquor to raise the pH to over 7. And when they did all of that, voila, the Nicardia went away. They have since added an anoxic zone up front to recover alkalinity, and they don't hardly ever feed bicarb anymore, but they have some on hand just in case. And the plant hasn't had Nicardia like that for a long time. That was a real success story. Just saying, you have to change. If you don't like what you grow, change the environment in the tank, and you will grow something else, and hopefully it will get better. Okay, so the next one is type 0092. This is a septicity filament. I took the picture of the 0092 in the wet mount up above, and because I always underestimate the amount of this one. This one usually you usually don't even see it, okay? And so you underestimate it until you do a gram stain or a Neisser stain. And the Neisser stain is the purple one, the one on the bottom right, and it tends to grow and get deep purple, and nothing else looks like this one. It's just a clear filament of purple. There's the gram negative one on the left-hand side, but the definitive stain is the Neisser stain. This one grows under septicity and usually under the breakdown of biomass. You have a clarifier full of solids, usually in a package plant that they don't have a whole lot of inventory control in their sludge, and those get a lot of mass, and it goes into the clarifier and it just sits there. And by the time the mixed liquor that comes into the clarifier gets to the bottom and gets returned to the aeration tank, it's gone septic. And when that septicity hits the aeration tank, you grow 0092. So the response, optimize your clarifier operation. Keep your solids down so it settles better, and so you don't have a clarifier full of solids. Same thing with your digester. If you leave the air off in your digester for a long time and it starts going septic on you, and then you decant that septicity back into your aeration tank, you're going to grow type 0092. Okay. Thiothrix, this is a classic septicity filament. It's big, it's readily identifiable, it's rectangular. You can see you can easily see the individual cells. It is gram negative. Usually it can have some gram positive reactions. And it also can, if you have sulfide in your waste stream, you can, it will take the sulfur or sulfide and convert it to elemental sulfur and include it in its cells, and it lights up like a Christmas tree there in a phase contrast microscope. Okay, Thiothrix is one of those ones that's hard to get rid of. They grow under septicity conditions, low molecular weight organic acids. When you hit that aeration tank with the solved oxygen, it will grow. If you have sulfides in the collection system, or if you're putting it in from solids handling, or your clarifier's putting it in, you'll see sulfur in the body of the filament, and it usually occurs when you have slightly higher F-M environment. Okay, so the response: remove your sources of septicity, and this can be something as simple as a long low-flow force main, or excessive clarifier sludge blankets, digester decants. And if you do some pre-aeration like say if it is a long force main that comes into the influent, that can sometimes help. Type O21N. This is one that is gram negative and Neisser negative, but the wet mount is so recognizable. Tony Glimp calls this a stack of hockey pucks. And that's what it looks like. They're shorter, they're wider than they are long, and so it looks like a hockey puck. And this will give you really bad settling. It's a very, very hardy filament and hard to get rid of, and it's grows all over the place. Okay, it grows on septicity, low molecular weight organic acids, and over a wide range of FDMs. Okay. Again, just like all the other septicity filaments, get rid of your sources. That's the hard part about septicity filaments. You have to figure out where the septicity is coming from. Most people think someone's dumping on them. I always say, do what's easy first. Look inside the fence. Look at your digester. Look at your clarifier. Look at your force mains. Okay. Much easier to fix something inside the fence than it is to search outside the fence. And anoxic selectors work for this one. Okay, if you just create a mixed anoxic zone, you give the flock formers first crack at that soluble carbon, soluble food. Type 1861, this is one that's, again, I very rarely see this one. But look at the filament. You can see in the bottom view, you can see how long the filament is and how narrow it is, okay? That's a pretty distinctive identifying characteristic. It's probably 5 times longer than it is wide. Again, this is a septicity filament, but it also grows under a lower FDM, not really low, but lower FDM. It's not very common in domestic wastewater. And again, remove the sources of septicity and maybe decrease the MCRT a little bit. Vegeatoa, this is one that's fun to watch. It's always lit up. Again, it takes sulfide, converts it to elemental sulfur and stores it. And under a phase contrast microscope, it will light up like a Christmas tree, like in the top photo. It's gram negative, Neisser negative. You can see some of the inclusions in the Neisser view, but really you don't need either of the stains to identify a Vegeatoa because it's the only filament that glides around. It's constantly moving and it's all lit up. It's very, very interesting to watch. Grows on sulfides in the waste stream in low DO, and you can pre-aerate to remove the sulfides in the waste stream occasionally. This one, you usually never ever see it in enough numbers to give you low or give you poor settling, but it is fun to watch. There's this one, Nostocoida limicola, hard to say, but this one can go either way. It can be gram positive or gram negative. You see the 2 reactions right there, but it is always Neisser positive. This one, again, looks kind of like the hockey pucks, but it's a little bit smaller than the type O21N, and it does stain gram or Neisser positive. Very distinctive Neisser positive reaction. You don't see this too often. It usually grows inside the filament, and well, let's see here. Septicity, wide range of FDMs. Oh, this is a nutrient deficient one too. This was one that can show up if you have nutrient deficiency. And you have to investigate the septicity and the organic acids. But also this one kind of shows up when you have food processing sources tributary to your wastewater treatment plant. That's one that I have seen in, I think it was the Apple processor, I believe, that they had a lot of Nostocoida limicola. Type O14 is, or O14, wait a minute, type 0914. Okay, there you go. Talking way too much here. This is one that is gram positive, Neisser negative. It has really heavy septa. That's the line between the individual cells. You can see that in the upper left-hand corner. And the other distinguishing feature of this one is that if there's sulfur or sulfide in the waste stream, it will store the sulfur as elemental sulfur, but they're stored as rectangles inside the cell. Everything else is stored as a dot, as a circle, a sphere, but these are stored as rectangles. And I've only seen it one time, and I tried to take a picture of it, and the picture didn't turn out quite so well, but those are rectangles. Again, another septicity filament with sulfides and low F to M. Okay, eliminate the septicity, eliminate the sulfides. Spiralis natans. This is one that you see fairly frequently. This is one that looks like sausages in a straw. In the upper photograph, you can sort of see right in the middle there where there's a gap between the quote sausages. And that is because, well, the cell's missing there, but that shows that it's got a sheath around it, okay? And in the bottom right photograph also shows the sheath in the one that's going towards about 4 o'clock there, sort of in the background. You can see some cells and then you see no cells. That's evidence of a sheath. Spiralis natans has false branching, which just has the two filaments sticking close to each other. It looks like it's branched, but it really isn't. There's no single cell that has a branch on it. And this is a low DO filament. And it can be low DO for the applied load, or say you have a real soluble load coming in and it's readily metabolized, and then the DO can get sucked up very, very fast. It might not be in the bulk liquid, but in the vicinity of the bacteria, you could have a low DO situation. And you can also have that low DO situation in the interior of the flock, because the readily metabolized organic food that comes in gets taken up, and as it's taken up by the bacteria, the DO doesn't penetrate into the interior of the flock. So the response, increase your bulk dissolved oxygen. You might have to increase it up way beyond 2 parts per liter to get it to diffuse into the flock. Type 1701, this is another one. It looks kind of like the 1851 that grows in bundles, but it doesn't always grow in bundles. And again, the picture on the right shows it looks kind of like the Spiralis natans, only much thinner, in that it has the sausage shape cells, and it is gram negative and Neisser negative. It could be mistaken for the 1851, which is gram positive, although weakly gram positive. But this is a classic, another low DO situation. It's a low DO, it has low DO, it will survive when there's low DO in the aeration tank, because it will outcompete the flock formers for the scarce DO, and it grows under a wide range of F to Ms. The response, again, increased dissolved oxygen in the aeration tank. Then there's this one, Haliscominobacter hydrosis. H. hydrosis is what they call it for short, because Haliscominobacter is hard to say. This one doesn't really give you much of a settling problem, but what it looks like is like pins in a cushion of flock. And it usually extends out from the flock, and it's very thin and usually pretty short, okay? And it's gram negative and Neisser negative. Doesn't really give you a settling problem, but you will see it incidentally in some places. I've seen it at one place in particular that had really high ammonia coming into a small package plant. High influent nitrogen is one of the things that will support it. Septicity, low DO, high influent nitrogen, ammonia, wide range of F to Ms. And again, increase the DO, remove the sources of septicity, and you should be able to select against it. Whew, okay, so coming up on the summary here. Now this is yet another chart. We saw it with the Nocardia forms in the Nocardia situation, but this one is one of the most valuable charts in that book, because what it does is it shows you the common filaments on the left-hand side, and it shows you where they grow in relation to an MCRT or F/M. And what it does, it separates the ones that you can waste your way out of from the ones that you can't waste your way out of. Now, if you can identify the filament, and it's any of those ones that go from type 0041 down to type 0581, where it goes from 8 to infinity on the MCRT scale, those are all filaments that you can waste your way out of, okay? The ones at the top with the really long arrows are the filaments that you can't waste your way out of. These are the septicity filaments or the low DO filaments, okay? So if you can identify the filament, you can look at a chart like this and it will tell you which ones to waste your way out of and which ones you can't waste your way out of. I think that's important. These are the same charts you saw before. Lots of red. Those are the ones that I commonly see. The rest of them are not only that I commonly see that, but will also give you settling issues. And the rest of them usually aren't very common or don't really give you very much of a settling issue. So there's like what, 7, 10 that really do cause, commonly cause settling issues. Remember, they grow under specific conditions: low F/M, low DO, oil and grease, septicity sulfide, and they can occur under nutrient deficient. Whew. Okay, so I'm about out of time. I'm going to go through staining real quick because staining is really easy and people are intimidated by staining, okay? So staining techniques reveal what is hidden, okay? Why stain bacteria? Well, because sometimes you have foam. Which bacteria is it? Which bacteria is that? We know which one that one is. What about that one? Foam? Those are all biological foams. So maybe you need to know which one it is in order to correct for it. Okay, so these are the filaments identifiable by staining: Parvicella, Nocardia, Mastocoida limicola, type 0041, 1851, 0914. Those are all gram-positive. The Neisser positives are Microthrix, Nocardia, Mastocoida limicola, type 0092. Okay. What do you need for staining? Well, you need a microscope. And you need a bright field setting, not phase contrast. You don't look at stain slides under phase contrast. Preferably you have a 100x oil immersion objective. These are special objectives that you can use immersion oil to bridge the air gap between the cover slip and the objective lens so you don't get a distortion. But you also need absolutely a minimum of a 20x objective to actually try to identify the filaments. Okay. Tools: you need microscope slides, and you need to air dry the sample on the slide. You need a clothespin. Why? Because if you don't, and you're holding the slide, when you stain the surface of the slide, you'll stain your fingers. So if you hold it with a clothespin with a spring on it, a spring clothespin, you won't stain your fingers unless you're really sloppy, like I am sometimes. You need a wash bottle to rinse. You need a watch with the second hand to time the reaction. You need some paper towels to blot dry, not rub, but blot dry the slide. And you need a sink, preferably a black lab sink, because if it's a white one, you're going to stain it, or a roasting pan, okay? and bone amine scouring powder if you are doing it in a hotel room like I have done before, and stain the sink. Okay. So this is a Graham stain. You have 4 fluids that you use: Graham crystal violet, Graham iodine, Graham decolorizing solution, and Graham safranin solution. And the procedure: Graham crystal violet for 1 minute, rinse with DI water. Graham iodine solution, you flood the slide for 1 minute, rinse with DI water. Graham decolorizing solution. This is the only tricky part to this. You hold the slide at about a 45-degree angle and apply dropwise from the top of the slide, and it drains down into your pan or sink until the blue color pretty much stops, and then you blast it with DI water to stop that reaction. Okay, because the decolorize, you can over decolorize it and it'll wipe all the stain off. Okay, and then you blot it dry, blot it, not rub it, blot it dry with a paper towel. Then you counter stain it. This is the negative stain with the Graham safranin solution, 1 minute, and then rinse it with DI water. Dry it again, and then view the slide 1000x or under bright light. No cover slip is necessary. Put immersion oil right on the slide. Okay, and you get this picture. In this picture, okay. Tools for staining for a Neisser stain. You have 3 reagents. You have methyl blue, which is solution A, Neisser crystal violet, which is solution B, and Bismarck brown solution. That's the counter stain. You need a transfer pipette and a little bottle for mixing the solutions A and B, and you have to make it up new every time you do it. And of course, you need a clothespin. So what you do is you mix 2 parts of the methyl blue with 1 part of the crystal violet in a small container. You flood this slide for 30 seconds. You rinse it with DI water. You don't dry it. Then you counter stain it with the Bismarck brown solution for 1 minute. Rinse it with DI water. Blot it dry. Don't rub it. And then view the slide at 1000 power. Okay. Bright light, not phase contrast. And you get the Parvicella stain and then the Cardia stain, or the 0092 stain. Okay. There I have 1000x. So why stain the bacteria? It's actually very easy. It will actually, on those certain bacteria that have the positive reactions, it will actually determine what is growing in your plant. Okay. If you're not really sure, looking at the wet mount, shows you what's hidden in the flock, like 0092 or 0041. You can't really see it inside the flock, but it will stain inside the flock, and then you can discover its relative abundances. And if you don't have a phase contrast microscope and you only have a bright light microscope, get a stain, and you might be able to still identify your filaments because you look at stained filaments under bright light microscopes. Okay. That's it. I'm only 10 minutes over, so. And I think I forgot to do my last polling question, didn't I?
No, we're good, John. We'll
Okay.
skip the polling questions because we have so many questions that Laura and I are going to ask of you, and
Okay.
we're ask you to be, and thank you for the wonderful presentation. I'll thank you right now. Slides are amazing. We have a lot of questions, so I'm going to ask.
Okay.
We're going to try and get the most questions done by being, if you can give the best response you can in a brief moment.
Okay.
And that way the people that have asked can also get their questions answered. So we're going to start doing this chronologically here.
Okay.
So I'll start with the first one. I'll let Laura cut in after. So I see something here about multiple growth of microorganisms in higher temperature, possible or not?
Multiple growth of
microorganisms in higher temperature, is it possible or not?
Well, yeah, well, actually in municipal wastewater treatment plants, the temperatures are pretty much follow the ambient temperatures. They probably lag a little bit. You know, I can see temperatures of like maybe 25 C, 26 C, Celsius. And the higher the temperature of the water, the more active the bacteria will grow up to a point. There are bacteria that are called thermophilic bacteria that grow in some high temperature conditions, like ATAD digesters or even anaerobic digesters that are heated. Those are different bacteria than what grow in the aeration tanks. So other stuff will grow, but they might not be exactly what you want. So okay.
I'm not going to give you much more time than that on each question. So
Okay.
Laura's got the next question, or I'll ask the next one.
Yeah, this question is, I normally take my samples for microscope from near our membranes after our pump. Should I be pulling samples from a different location?
Actually, if you have a membrane plant, you probably have an anoxic zone too, but I would take the sample just from the bulk liquid. It is surprisingly uniform. I can take samples from all over a tank and they will pretty much have the same bacteria in each of those samples. So I don't really think it matters. I usually take it at the end of the aeration, so that would be near the membranes.
So I'm going to keep cutting in because we want to get through as many as we
Okay.
can. If aeration water color, it's become brown to turn pale, seems like to turn pale brown. Is there a problem?
There might be if it's a change, a sudden change. It could be that you have I&I, or you might, if you're close to a river, you might have river water coming in, or you might have dirt or construction, leaks in your sewer. You can see silt under a microscope. Yeah, I don't know. That's a good question. If it's starting to go septic, it usually turns even darker. If you have a washout, it might be lighter. So that's a good question. I can see how it can change, but nothing real definitive.
Okay, Laura.
This question is, do you have a range of TSS that you would consider ideal, as in not too much mass?
Okay, I covered this in my package plant show. You need to have enough mass so that the ammonias are low, conversion is complete, but not so much mass that separation is impacted. So what you do is you do a settleometer when the ammonias are low. If it doesn't settle very well, you do a diluted settleometer, and that would be like if you waste half your mass and see if it settles better. If it does, then you probably need to waste more. So those are the two criteria: good settling and good conversion.
Okay, John,
It's going to vary for every plant. Go ahead, next.
So now, again, this is early in your presentation, this question. Is that a BX40?
Yes, that's my microscope. That's my baby. Yep.
Okay. That was quick. I like the quick ones, so we get
Okay.
everybody.
That's a great microscope. Yeah, go ahead.
The next question is,
trying to leapfrog each other, Laura and I, so we'll see if she has something or I'll find something.
Okay. What's the best temperature to store mixed liquor samples for microscope analysis?
Well, it depends on what you're looking at. If you're looking at protozoan, you should probably get a fresh sample. I have operators send me samples through the mail, and if it's filamentous, the filaments will stay, but the protozoans will likely die unless you leave an airspace and it gets agitated as it's being delivered by the mail. So anyway, I don't really store it, but room temperature, I guess. Maybe slight refrigeration if you're keeping it for a couple days. Filaments don't go away. Yeah, so go ahead.
So moving forward, I'm going to cover two things here.
Okay.
People are asking about the handouts. So if you're
Mhm.
having trouble downloading them or something, just get back to Laura, myself. We'll try and see what we can do about sending the handouts to you. But they are in the handout section, and you can be downloading them now. And we'll leave the webinar open a little bit after we're done here. So here's the question: which book is your go-to book? They said they missed your response on that.
Okay. The go-to book was the one in the middle. It was Jenkins, Richard, and Dagger. Causes and Control of Activated Sludge Bulking. It's a really long title, and I can never remember it, but it's the orange covered slide. That just has so much information on it.
Okay.
But the other two are good as well, but that's the one I really like.
Okay, great. Laura?
What do bristle worms indicate about the sludge environment?
You're probably not wasting them enough. To grow, like Dr. Richard said, if you have worms in your mixed liquor, you've gone beyond activated sludge and you're now at liquid compost. And he meant nematodes in particular, but bristleworms are compost organisms. They're higher organisms, and you probably just need to waste more.
Okay, I'm moving forward if that's
Sure.
good. Do you have a range of TSS that you would consider ideal, as in not too much mass?
We already answered that question.
Oh, I'm sorry. Did I miss that?
Yep.
How do
Whatever
you?
settles well. Whatever settles well. I don't
Right.
do spin solids analysis. I do a centrifuge spin, and I do centrifuge spin if it's less than usually about 3. Above 3, you might have a settling problem, but under 3, you almost won't have a settling problem unless you have filaments.
Laura, I'm going to take this one unless you have it. How does filament develop quicker than the flock?
When nutrients are scarce, filaments will outcompete the flock because they have more surface area, and bacteria absorb their food through the surface area. Okay, so the filaments, when resources are scarce, will outcompete the flock formers. If you give them an anoxic zone, the flock formers will get first crack at the soluble carbon and will outcompete the filaments.
Okay. Laura, if you're good.
What numerical range are you considering low FM?
You know, I don't consider a number for that. There's problems with the food calculation. There's problems with the mixed liquor calculation. I mean, the food calculation is a BOD test, which are plus or minus 30.5 points on a standard. And so that one I think is too variable. And the mass calculation, and if you burn it, they figure that's what's living. Well, hair burns, lint burns, all sorts of stuff burns. Concrete will even burn if you put it in a 500-degree furnace, so it doesn't burn, but it loses moisture. So I think an FDM number doesn't work. If you have too much mass for the amount of food coming in, you have a low FDM environment, and there's not a number associated with that.
Next question.
You would just grow low FDM filaments. Okay, next question.
How does a single-celled organism have different parts?
Well, they have structures. It's clear in a stalk ciliate, you can see all those structures inside of it. Bacteria has inclusions. They have DNA and RNA, I guess. Those are all structures too. So, I'm not a microbiologist, okay? So I'm an engineer that has a great microscope and has made great use of it. But the ins and outs of microbiology, you'd have to ask a PhD that one.
Okay, we'll move on.
Does your laboratory receive commercial samples for identification? If so, would this information be available online?
I don't have a lab. I am a state employee that has a microscope, and I go to wastewater treatment plants with the microscope in the back of my pickup truck. I use it as an investigative tool to help me diagnose settling problems. And I've been looking at microscopic examinations for well over 20 years. And, you know, what I do is in my presentation. I mean, I have hundreds of pictures of mixed liquor clogging up my computer memory, but yeah, I don't know.
All right, we'll move on.
Sure.
And if I'm moving on too soon, John, let me know.
Yeah, that's fine.
How often do you recommend operators document protozoa?
Protozoa are interesting to watch. They're charismatic. They demand your attention because they're big and they move around frequently, or they're interesting to look at. But really, they don't do the work. The bacteria does the work. And so really what I focus on are filamentous bacteria to tell me what the conditions are in the aeration tank, and the flock structure to see if it's going to settle or not. Protozoa are interesting if they're dead, I note that, or if they are higher organisms versus lower organisms, or real low organisms, I note that. But I usually send pictures out when I do an analysis and a report on it. But protozoans are there because their food source is there, and but really it's the bacteria that do all the work.
Okay, Laura.
You mentioned nematodes being part of a composting system, but then you also mentioned that you may not be wasting enough. Is having too many nematodes a problem?
I don't necessarily think it's a problem. I usually don't see that many of them. I will see some of them in a slide view. I think that was just a pet peeve of Dr. Richards that talked about being compost organisms instead of activated sludge organisms. I see them frequently, but I don't think they cause problems necessarily.
So I'm just going to let the audience know that we're going over because there's some great questions here, and that we'll turn the recorder off at 3, but we can go on as long as we want with the questions after that for those that want to stay on. So the next one, what's the best temperature to store mixed liquor samples for the microscope analysis?
I would say probably just a refrigerator, but you know, when I am going to a plant, I just go out and grab a sample. I grab it right fresh and take it right in and look at it. Like I said, I have operators send me samples through the mail, and only rarely is the sample unfit for looking. And even if you get some spirochetes and stuff in there, which is sources which indicate septicity, the filaments will be preserved. They're not going to change that quickly.
Great, thanks, John.
Is there a scenario where unfavorable microbiology will grow when the DO is too high?
When the DO is too high, usually, well, let me think about that. Maybe if you have low FDM filaments, but DO, it would just be incidental. What happens with really high DO is you might get flock shear. So you have a good sized flock, but if there's no food coming in and you have a really high DO, the flock, the air might shear the flock into small pieces, and it would give you turbidity and might accumulate as a suspended solid going out. That would be the only situation I can really think of. And you're wasting energy, of course. So that would be my thought on that. Too much DO doesn't really help you. You're just using up electricity. So for your blowers.
Can you recommend, I don't know if you did this already, John, you recommend a good phase contrast microscope in the $500 to $1,000 range?
I don't think you can get a good phase contrast for $500 to $1,000. There are lots of vendors that sell cheap ones, but the optics are so cheap. I've had operators get these, and I've tried to zero them in and get them to work, and the optics are, you get what you pay for with optics. Yeah, I think you'd have to spend at least maybe $3,000 for a decent phase contrast microscope.
Great.
Can you simultaneously both waste more and increase DO?
Yeah, probably. I mean, they're two separate operations. The DO is a function of the load coming in and how much aeration you're giving, and wasting sludge is what settles in the clarifier and gets wasted to the digester. They're two independent operations. Hope that answers the question.
So John, the next question I goes, says, not sure how this is in reference to what, but okay, so foam trapping is opposite of wasting foam out of the plant. I don't know if that's a comment or a question.
Well, if you don't trap foam, then low levels of foam that you might create will flow to the clarifier, okay? If you trap foam, it just keeps accumulating because it's on the surface, and if you have a subsurface takeoff from the aeration tank to the next treatment unit, it's just going to remain in play, okay? That's where foam trapping is a problem. The best way is to not have foam trapping so it doesn't accumulate, but then adjusting the wasting and the pH so that you don't grow the foaming filament.
Okay, great.
What composes the slime layer? Is it polysaccharides?
Yeah, it's lipopolysaccharides. It's sort of like a fat, well, let's see, a lipopolysaccharide, fat and sugar, I guess, something like that. But it's when their cell walls or cell membranes have a nutrient, it's missing either nitrogen or phosphorus, and then it creates this lipopolysaccharide slime, which coats the cell, and it gets really bad. If it gets really, really bad, you'll get slime bulking, and it looks just like a filamentous bulking, but there's no filament involved. It's just this. It's just the slime, and the fat part of it will keep it floating and buoyant. So I've only seen it a few times, but it's you can do a reverse India ink stain. Just put a little water-soluble India ink on the mixed liquor and then put a cover slip on it. And if the India ink doesn't penetrate the flock and it has sort of a bluish tinge to it, you probably have slime bulking. Does that make sense?
Hopefully.
Yeah.
So you mentioned, I don't know, excuse me if this has been answered. You mentioned nematodes being part of the composting system.
Mm-hmm.
Congratulations.
We answered this question already, Peter.
Yeah.
Oh, sorry.
I think we've gone over all the good questions that we had, so we should probably wrap it up.
Okay, we're gonna.
Can I say one thing?
John,
can I say one thing?
Yeah.
Okay, my contact information is on the screen. Feel free to send me an email. You know, I have an iPhone. If you can text that phone number, or if you really want to talk to me, you can call that phone number. but and the email comes into the phone as well, so, or my laptop. So any further questions, feel free to send me an email.
So John, we'll end the webinar recording now.
Okay.
But then we can, I'm stopping it now, and thank you very much. I want to say that it was excellent presentation, and people are congratulating on the quality of the presentation, and I'm going to stop